Hi everyone, my name is Tonka and I have prepared a presentation about erythrocytes. So, erythrocytes are known as the red blood cells, which are the most numerous cells in our blood, and they contain about 4 to 6 million of per cubic millimeter in our blood. Every red blood cell contains about 250 million molecules of hemoglobin with ability to bind to oxygen, which make them very important in transporting of oxygenated blood from heart to all body and peripheral tissue. They also carry about 23% of the blood's total carbon dioxide back to lungs to get the blood enriched with oxygen. Hemoglobin is a protein which consists of 4 polypeptide globine chains, and each chain contains heme with iron. Each hemoglobin can thus carry up to 4 oxygen molecules. They are shaped like a flattened discs that are intended on each side to maximize uh, the oxygen that can be carried on the surface area, and they are also very flexible, so they can squeeze through capillaries or one erythrocyte at a time. So there are different counts and measures we consider when we are assessing health status of individuals, and the most important is the red blood cell count, which is for male between 4.4 to 5.5 and for female 3.9 to 5.1 trillion per liter. Oh, this red blood cell count let us determine the production efficiency in the bone marrow for replacing old aged or erythrocytes which are depleted or lost in the blood, and they are being substituted with new cells. Though we expect around 1/4 of population to be anemic, meaning that they don't carry enough oxygen or the oxygen carrying capacity of the erythrocytes is impaired. And this can be due to the inefficient production of the red blood cells or excess blood loss, increased destruction of erythrocytes, or inherited blood disorders. And symptoms for anemia include usually shortness of breath, fatigue, or increased workload on our heart, as we need to pump more blood to get enough oxygen to tissues, dizziness, or high resting heart rate, which is causing the workload for heart bigger. Hematocrit is another number that represents the percentage of erythrocytes in the blood, and low hematocrit may indicate inadequate production of red blood cells, the normal production of smaller cells than normal size, or an excessive loss in bleeding, for example. And high count could be due to poor removal of red blood cells or an excessive uh, production or dehydration. Men should have between 51 to 53% and women between 36 to 46% of all blood volume filled with erythrocytes. The mean corpuscular volume, MVC, shows the average size of red blood cells, and low can indicate anemia caused by deficiency in iron or chronic inflammatory or inherited condition. And high MVC indicates macrocytic anemia, which is caused by deficiency in vitamin B12, or it can be caused due to toxicity from alcohol or drugs. The red blood cell distribution count width is a measure of the variation in the size of erythrocytes and is very useful to help determine the cause of the anemia. Mean corpuscular hemoglobin concentration is another measure, and that indicates the average concentration of hemoglobin in erythrocytes. It is very important when we are diagnosing anemia, as we can see exactly how much hemoglobin is in each erythrocyte, or like average numbers. And the low values usually indicate inefficient production of hemoglobin in erythrocytes, and the high numbers are usually caused by vitamin B12 or folate deficiency and certain types of anemia. And the last count I mentioned is reticulate count, which stands for the percentage for newly formed erythrocytes that are still in the process of maturation, and they make up usually between 0.5 to 2.5% of total red blood cell count. And we produce about 2 million of reticulocytes every day, and they travel in the blood for about 2 days before they mature. Erythrocytes also have a hormonal feedback function in our organism, since the low oxygenated blood stimulates erythrocytes production at the bone marrow, which is caused by the enzyme hormone called erythropoietin, which is produced in kidneys. And this hormone then stimulates the bone marrow to produce more erythrocytes. And so we are getting to the formation. So erythrocytes are produced in the bone marrow and released into the bloodstream, where they function as carrier of oxygen for about 120 days, and then they die and they are reutilized. So red blood cells are formed from the hematopoietic stem cells, which take place in the bone marrow through framework known as hematopoietic niche, where a series of differentiation steps take place, highly depending on the environmental factors. After the first stage of maturation, when we see erythroblast enucleation, which is reticulocyte type 1, also known as R1. It is a very young reticulocyte that contains necessary elements for the synthesis of hemoglobin, and that's one thing that differentiates it most from other stages. And it has a spheroidal shape with the surface strongly invaginated and with the roughness created by the nuclear extrusion. And billions of erythrocytes are generated every day from megakaryocytes, with differentiated process led by erythropoietin, the enzyme produced by kidneys. And during the process of differentiation, cell undergo morphological changes. We can see in cell size, which we can see the cell size is being reduced, and chromatin condense, with increased production of hemoglobin, of course. The cell lineages faces one crucial final step phase before they mature, which is expulsion of the nucleus from erythroblasts, which lead to the formation of reticulocytes and the loss of organelles and ribosomes through autophagy exosome combined pathway. Erythrocytes production depends on the composition and stiffness of the extracellular matrix as well, as it can model cell behavior through mechanical and chemical signals in our body. So erythrocytes live around 120 days, and they function as bringing oxygen into our tissues in our body and take carbon dioxide from tissues back to lungs, and then it's being diffused and oxygen is again being bonded to erythrocytes, and that's how the cycle works. Erythrocytes are able to deform to perfuse small capillaries in which vessel diameter reduced to only a quarter of the cell's resting diameter. Cellular deformation also aids in large vessels, not only in small capillaries, and there it's with the cell orientation and alignment with the flow and its ability for the cells to migrate toward the central region of flow, and they also reduce viscosity of the cell. And the death, so process of erythrocyte aging is characterized by the expression of death after progressive biological and chemical changes, mainly due to reduction of metabolic machinery involved in membrane vesiculation, hemoglobin modification, deamination of protein, decreased deformity to perfuse through capillaries, and progressive failure of both cellular homeostasis and antioxidant defense. All erythrocytes are tagged with specific antibodies for removal by macrophages from bone marrow and die in spleen, liver, and bone marrow. Erythrophagocytosis is triggered through the oxidant stress. Every second, there are 2 million erythrocytes that die, which is the highest turnover of any cell type in the human body. Erythrosis is a form of cell death by phagocytosis, and is triggered by toxins, drug, disease, oxidative stress, or hyperthermia, and this process is triggered by an increased intercellular calcium concentration inside the erythrocytes. Its structural compositions are then recycled and produced in new erythrocytes in splendid form of renovation, such as heme containing iron. In hereditary hemolytic anemia, cell deformability is almost always impaired, and then the majority of abnormal erythrocytes are retained and destroyed by phagocytosis. So that's why it's called hereditary anemia. So thank you for watching, and here are my sources.